All right, so thanks so much for the introduction, and it's really cool to be here. This ended up being, this is my first time coming to this conference, and it's super cool, really interdisciplinary. It's awesome to see how people are approaching brain networks from different scales and so forth. And so I guess I'll be adding to that from taking a little bit more of a zoomed out angle and a little bit more of a clinical angle. So I'm Jake Vogel. I'm an assistant professor at the university. And I do study neurodegenerative diseases, and networks are a big part of what I'm doing. And today I'm going to be talking to you mostly about these connectome-based models that we've been using to better understand neurodegenerative diseases, and also hopefully have some sort of application that's useful in clinical purposes. So the overall sort of outline of the talk is I will just sort of describe what I mean by these connectome-based models. I'll talk about how we've been trying to use them to learn new things about Alzheimer's disease, and we are trying to push it into clinical applications as much as possible. So I'll cover kind of what we've been doing there. If I have time, I will talk a little bit about sort of more dynamic network changes over the course of Alzheimer's disease using more sort of traditional fMRI approaches. But I think the bulk of the talk is going to focus on these connectome-based models. And I will also really be focusing today more on application. So I'm not going to throw too many equations and stuff at you. I really want to focus more on, what we're using these models to do, because that's sort of what's more interesting and important to me. Hopefully you'll feel the same way. Okay, so let's start with sort of like the background of what these connectome-based models are and what they do. So, back in the old times, really we just had these clinical syndromes, and I think there's a lot of parts of medicine that are still here, right? You just have a bunch of symptoms and you don't really know necessarily what's causing it. And even though now we know a lot more about neuropathology, the fact that we started with clinical syndromes is still really reflected in the medical terminology, because we really just describe most diseases based on the clinical syndrome. But of course, we sort of learned that there are certain neuropathologies that lead to these specific syndromes that cause dementia. So that was great. We were like, okay, great. We have neuropathology. Someone comes with a syndrome, and we can know, okay, they've got this neuropathology. We can learn to treat that. We can learn to recognize it, and we're golden. But the problem is the picture is actually really, really complicated. There are various types of neurodegenerative pathologies. They're not even necessarily all listed here. They can lead to various different symptoms, like you can have, for example, frontotemporal dementia from TDP-43 or from tau. So the picture is very complicated. So there's kind of, we need to find these like intermediate phenotypes between pathology and clinical syndromes. And so that's sort of like where I'm coming in here. And but of course, as all of us are, we stand on the shoulders of giants. Oh, sorry, I forgot to mention that there's a little bit of a kind of an advantage that we have. When we look at the sort of distribution of all these different neuropathologies in the brain, we see interesting patterns, right? It's not like there's just pathology everywhere, right? We have each of these different neuropathologies and these different syndromes build up in very specific patterns, and they sort of start in usually very focal regions and then sort of spread through the brain or propagate through the brain. And that's a very interesting clue as to sort of what's going on. And so way back in the 80s, there were already neuropathologists who were looking at slides and sort of coming to the conclusion, and I'll sort of quote here, they support the suggestion that the pathological changes in Alzheimer's disease affect regions that are interconnected by well-defined groups of connections, and that the disease process may extend along connecting fibers. So already in the 80s, there was this idea that possibly the way that neurodegenerative pathologies spread through the brain is actually by using the brain's own connectome architecture, and really using the connectome as a circuit to essentially almost like highways to spread through the brain. And so there was a really, really amazing study in 2009, maybe some of you are familiar with it, that really, I think, brought this idea to the fore. And it's really since become a major hypothesis in neurodegenerative diseases. So here, this was a study by Seeley and colleagues in 2009. And you can see here, this is a VBM maps of 5 different dementias, right? So Alzheimer's disease, frontotemporal dementia. This is semantic dementia, which is sort of has a primary language deficit. Primary progressive non-fluent aphasia, another type of FTD, and cortical basal syndrome. And they all have very different syndromes. Like this is more of a motor, this is more amnestic, and so forth. And these are basically atrophy maps. So it's where these people with these types of dementia have brain atrophy compared to controls. And so what Seeley did is they basically found the sort of area of peak atrophy in each of these dementing syndromes. And they used intrinsic functional connectivity from healthy young people to basically look at the brain networks that are associated with these areas of peak atrophy. And that's what's shown here in yellow. And what they observed is that the sort of, if you seed these regions of peak atrophy, you get these functional networks that look very similar to the regions that are atrophied in these different neurodegenerative diseases. And so what they hypothesized based on that is actually brain atrophy is really traveling through intrinsic brain networks that you can already measure in young people. And this was really, I think, a monumental hypothesis or a study, because it really pushed this hypothesis that neurodegenerative diseases involve basically spread of degeneration through neuronal networks, and neuronal networks that are specifically involved in certain types of cognition and behavior. So this kind of posits brain networks as a sort of intermediate phenotype between neuropathology and syndromes, which is really great for us because, as a neuroimager, this is something that we can measure really well. We can measure brain networks well, and it's actually pretty hard, I think, to measure brain networks, for example, like macro scale brain networks anyway, in a lot of the other disciplines. So we're sort of like trying to take advantage of that and really understand how these brain networks contribute to neurodegenerative diseases. So if we take Alzheimer's disease, which is the main focus of study in my lab, you have these actually two different neuropathologies, right? You've got amyloid pathology, which builds up really actually all over the brain very quickly, very early in the disease. And you can see actually by the time you have symptoms, your head is already completely full of amyloid, essentially. So you can actually have a head full of amyloid and have no symptoms at all. It's very, very early and really very diffuse. Tau is the other pathology. And I think it's something that we're more interested in for several reasons. One, it builds up very focally in one region and then spreads in a rather stereotyped fashion through the brain, which is really compelling and interesting. But also the spread of tau is much more closely associated with cognitive decline and with neurodegeneration and brain atrophy. And luckily, we can actually measure both of these neuropathologies in living humans with PET. So since about 2004, we can measure amyloid in the brain. And since 2015, we can now measure tau in the brain as well. And it's really been able to push the field forward. So coming back to this hypothesis about, does pathology spread through the brain? This has undergone a lot of sort of study over the last decade. And so the theory is that you have this tau that's building up sort of in the cell body. Monomers or possibly oligomers spread actually down axons, end up in synapses and actually somehow spread across the synapse into the postsynapse of another neuron. And by doing that, again, the neurons are almost being used as conduits for the spread of pathology through the brain. And so if you zoom out, if you think about how this would look from like a macro scale perspective, it means that regions, if you have a sort of epicenter, let's say here in the entorhinal cortex, if you have areas that are more connected to this epicenter, you should see more pathology there sooner. And if you have an area that's sort of less connected to the epicenter, you should see less pathology there sooner. That's great because that creates a framework that we can use to study with neuroimaging, right? And so this is what we do with these connectome-based models. So we can sort of create, if you like, a sort of artificial brain system where you have these different nodes, which are essentially brain regions, and you have these edges or relationships between the nodes. And so here we can use functional connectivity or diffusion-weighted imaging or whatever you'd like, basically, to represent connectivity between nodes. And of course, within the nodes, we can represent neuropathology based on our objective measures of either brain atrophy or, in this case, PET, which lets us actually image, the location and amount of brain pathology in these different regions. And if we do that, we can come up with fairly simple models where we just look at the connectivity, for example, to an epicenter and the amount of pathology in a region, and we can really correlate those things and ask, is there a relationship between connectivity and the kind of pattern of brain pathology in these different diseases? And so we like to take that a little bit a step further. And instead of just doing sort of correlations, we do simulations. So again, you've got your same kind of artificial brain network where you've got brain regions, and you have use sort of normative connectivity as a measure of the underlying relationship between these regions or connectivity between the regions, I guess. And we can just put a signal in the entorhinal cortex or some other epicenter and let that signal diffuse through the network. And once we've let it diffuse over a certain amount of time, we can compare this simulated diffusion to the actual observed neuropathology that we see using PET. And if we come up with a sort of solid relationship, that provides pretty good evidence that there is actually some network spread going on of neuropathology. And you can do that also at the individual level. You could do it at the group level. There's a lot of options there. So we have, way back in 2020, we applied this to some PET data that we had from ADNI in the BioFinder cohort, and we basically got a really, really good correlation. So here is the sort of, each one of these dots is a brain region. This is sort of the predicted tau in that region, and this is the observed tau in that region. And you can see that just by simulating the spread of pathology from the entorhinal cortex into different brain networks, or really through the brain's network, we can actually recapitulate the overall pattern of tau in the brain in Alzheimer's disease really, really well, like surprisingly well. So we feel like this really supports this hypothesis. What's also nice is we actually tried to use different epicenters. We actually used several different brain regions and reran the model. And what we found is that the best models always use the entorhinal cortex. And again, this is really important for us to validate these models because if this is the sort of Brock staging system, so this is the staging system that we use to progress tau pathology in Alzheimer's disease, and you can see the very first brain region that we see tau pathology is the entorhinal cortex here, and that is the best epicenter for our models. So it really seems like this model is supporting this notion that tau pathology spreads from the entorhinal cortex through anatomical connections through the brain. So this is really cool. And many, many other labs have now replicated this. This slide is actually probably about 2 or 3 years old. A lot of other people have replicated this since. It's really, really reproducible. And so we learn a lot from this, right? So we learn that, it does seem that in Alzheimer's disease, tau neuropathology is distributed in patterns that resemble brain networks. That's very interesting. Connected brain regions are more likely to show pathological load. All of it supports this idea of this network spread hypothesis in at least Alzheimer's disease, if not other neurodegenerative diseases. And so this has actually led to a legacy, right? So besides all this great neuroimaging work that's been going on over the last decade, there's also been a lot of really excellent animal work that's basically been trying to see if this is feasible, if it's possible in living animals. And they've done pretty much everything. They've basically shown that, yes, you can get monomers to spread down axons. The neurons will uptake tau at the synapse. They will also secrete tau when they depolarize. So a lot of the mechanisms are shown to be possible. And at the same time, this is already leading also to drug development, right? So there are, at this time, several different existing drugs in the sort of pipeline that are really geared at stopping the spread of tau. So not necessarily even stopping the production, but really trying to stop it from spreading from region to region. And there continues to be a lot of really nice biological work that's trying to understand the mechanism by which this happens in humans and in animals. So I think this is really cool because it's not that often that in neuroimaging where we're really zoomed out and we don't have really solid, always hold on microscopic biological mechanisms, we don't always have an opportunity to contribute to the sort of understanding the biology of a disease. And here it really seemed like the neuroimaging community was contributing a lot. And so I always find that really cool. Okay, cool. So that's so now great. So now you have an understanding of how these connectome-based models work and why we like them, why we think they're interesting. But we think that we can take this a lot further, right? Because we essentially have a simulation of Alzheimer's disease in a way, or at least some part of it. And so what can you do with that? So one of the things that you can do is you can try to learn more about the disease, right? So we're very inspired that we've sort of been able to contribute a little bit to understanding of a mechanism in a disease. And so we want to push this a little bit more. Can we use the sort of tools at our disposal to learn even more about the disease using connectome-based models, right? So going back to this pattern that we see in Alzheimer's disease, right? This Brock staging system. So usually when we use tau PET, we can stage people based on their what stage they're in using PET, but usually we just look at the total sort of amount or magnitude of tau in the brain. And when you do that, it actually doesn't really follow the Brock staging system. So this is just the basically if you take about 600 people that have tau in the brain and you average the amount of tau in each of these Brock stage regions, right, 1 through 6, the areas with the most tau is actually in Brock stage 4. So that sort of there's this idea that the sequence that tau builds up and where tau is in the brain are kind of these two different aspects of its distribution. And so this kind of makes sense, right? So here's 3 different cases of people who died with Alzheimer's disease, and all of them have at least a little bit of tau in this area called the inferior lateral temporal lobe, and that means automatically they are Brock stage 4. That's how we measure it. It's like if the tau has gotten to a region, then they are that stage. But you can also obviously tell that these people have different amounts of pathology in the brain, right? These two patients seem to have way more tau in the brain compared to this patient, and so this kind of belies this idea that Braque stage is a measure of tau progression, not necessarily tau load. So we want to be able to measure both progression and load because we think that these are both important for understanding the progression of Alzheimer's disease. And so we can measure load really easily. This is just by using the amount of signal in a tau PET scan. Great. To get to sort of this tau progression or tau sequence measure, we turn to sort of this approach we use a lot in the lab called Gaussian mixture modeling. And it works really well because even if you don't have any tau in the brain, it's PET signal and you're still going to get PET signal, right? But that PET signal is actually going to be fairly sort of distributed in a kind of Gaussian fashion because it's just noise. So it's got this nice normal distribution, and it's really easy to kind of pull that noisy distribution apart from the true distribution, which is much more skewed. And so we can use that to derive, using the posteriors of these Gaussian mixture models, we can use it to derive a probabilistic sort of measure of whether tau is in a brain region or not, just based on the value there. And that's really useful because when we do that, if we basically say, for every person and for every brain region, we probabilistically assign whether there's tau there, and we average that across everyone, we get this beautiful progression that now looks like it goes really nicely along the Braque stage regions, basically saying that the way to interpret this is about 80% of people in this cohort had tau in Braque stage 1 regions, but more like 20% had it in Braque stage 6 regions. So when we do this, it's really nice because we can have measures of both tau load and tau presence. Okay, so now that I've sort of gotten that little introduction to these different measurements out of the way, we can talk about how we try to explore these two things using connectome-based models. So we've been using a new model recently that I think is super excited. This has been adapted from our colleagues at McGill, Branislav Misic and Alan Dagger. They had this nice SIR model that they developed, and this was adapted by a postdoc in my group, Xiao Yu. And what's really cool about this model is it does the same sort of connectome-based diffusion as the other models that I was describing before, but it also tries to simulate some other sort of mechanistic things that are happening at the level of cells, or really cell populations in this case, which is also the synthesis of tau and the clearance of tau and the misfolding of tau and into sort of abnormal tau, right? And so it's kind of simultaneously simulating these properties inside of a brain region, and it's also simulating the diffusion of tau sort of and spread through the sort of brain network in the same way. And what's really cool is that we can take some of these parameters and actually we can use a priori information to modulate them, right? So we can say, I want the regional synthesis rate to be higher in this region compared to this region because I know that there's more X in that region that I think will affect kind of the spread of tau. So this is, I think, a really interesting model for us to kind of probe the mechanisms of what's going on in the disease and just see if it basically leads to better models. So I'll give you an example here. So this is basically just a straight replication of the 2020 paper I showed you earlier, where we look at this kind of measure of sort of tau sequence or tau progression. On the x-axis, we simulate just the spread of pathology through the brain networks, nothing special else going on there. And we end up doing a decent job explaining the overall pattern of tau pathology, especially really in these kind of early stage regions that have a lot of pathology to begin with. But if we try to do that same thing with tau load, the model doesn't do nearly as well. So this connectome-based diffusion, it's doing a pretty good job explaining sort of the sequence by which tau accumulates through the brain, but it doesn't do as well of a job explaining how much tau ends up accumulating there over time. So we can now try to understand these models better. So what we do is, using the Allen Human Brain Atlas, we can have actually a measure of the expression of this gene called MAPT, which is what encodes the tau protein. And we can say, okay, well, we actually know that there's more MAPT expression in different regions. So what if we allow the model to synthesize tau faster in brain regions that have MAPT? So just by adding that into the model, we actually are able to recover a lot of the overall progression of tau in the brain. And you can see we get a sort of much better score in terms of the predicting the regional distribution of tau through these diffusion models. We also get slightly improved models for tau sequence here, but sort of not to the same extent. So we thought that was really cool. We were like, all right, this was like a hypothesis we had. It was a really obvious one. And we applied it to the model, and it seemed to do a better job explaining the data. So we're like, that's cool. Maybe this model works. Maybe we can keep pushing the envelope a little bit. And we have this now enormous, really over the past like 5 years, we had this like enormous armamentarium of spatially resolved information in the brain that we can tap into, right? So a lot of this was curated by, again, Bratislav Misic's group at McGill, but they've really collected sort of spatially resolved information about the brain across many, many different sources. So now we have a set of really maps of neurotransmitter receptors and transporter sort of across the brain, different aspects of morphology and metabolism across the cortex, cell type distributions, right? We know where amyloid beta in Alzheimer's disease, it's distributed. We've got all aspects of microstructure. We have all these different things that we can turn to help us understand different properties of different brain regions. And we said, we're a computational lab. Let's just throw all of this at our model and just see if any of these properties end up explaining the distribution of tau better. And if it does, maybe we can start to kind of pay more attention to it, right? And we thought, we've also been really testing this hypothesis that tau is spreading through anatomical connections, but there's also, I'm jumping ahead. So when we did this, when we applied all these different approaches, no, I'm gonna skip this. So we also decided that we've been using anatomical connectivity this whole time. We can measure relationships between regions in many different ways, right? We can also say, for example, the covariance in gene expression. Why would we look at that? Well, instead of the idea that tau is just spreading through, anatomical connections, we also know that regions that are connected also share a lot of properties, right? They develop together. They have actually similar molecular properties. It's also possible that there's just a sort of molecular gradient of vulnerability that this pathology is cascading over. And so we're just, we're able to test different hypotheses with these different brain networks. And so again, we just threw all of these different properties at this model and just said, what are the best models? So I'm going to show you the results. It's going to be very busy because I'm showing you all at once, but we're going to walk through it slowly, I promise. So this is sort of like all of the results of this model, right? So on the x-axis, we've got different connectomes. On the y-axis, we have the performance of the model. Each dot is a model, and they're sort of colored by these different properties. Each one of these dots is a model with different properties sort of enhancing the model, and it's colored based on whether it's enhancing the spread or the synthesis or the misfolding and so forth. Obviously, a lot going on in this figure, but I'll show you sort of what we sort of take away from it. The one thing is that when we look at how well, when we add this regional biological information to the model, and we see how much does it improve the model over just using the brain's connectome to simulate the spread of tau, we find that it really doesn't give us much improvement in terms of the sequence of tau. Only when we look at the sort of overall load of tau in the brain do we start to get any advantages over this connectome-based model. And what this teaches us, we think anyway, is that the sort of brain network, no matter how you measure it, is a really good way of simulating sort of where tau is in the brain. It really seems like where tau distributes in terms of its sequence, it does seem to follow brain networks very closely, really almost no matter how you define those networks. But if you want to get sort of an idea of how much tau is going to build up at a region, in a given region, you also need to take into account these local properties. And so we think it's kind of really cool by combining these two pieces of information, we can sort of get both. Now, when we look at sort of the models that perform best, it actually ends up being these conventional factors that we would have expected from the beginning, right? So again, this MAPT thing that I showed you before, we also found that regions that have amyloid beta, the other neuropathology in Alzheimer's disease, tends to have a faster spread into those regions. And we also see things related to higher regions that have more clearance from the brain. They show sort of less tau. And so all that's really, really interesting, and it works across the structural covariance or structural connectivity matrices. So it really all supports what we've already expected about Alzheimer's disease, which is great. It gives some credence to our model, and it lets us know that we can use these models going forward. But when we look at sort of tau sequence instead of, or distribution instead of sort of tau load, there's a bit of a different story that emerges. And it's kind of interesting vis-a-vis some of the other talks that we've kind of saw today. And so I'm summarizing the information here across all of these models so you can see what's going on with different connectomes. And so as expected, these structural connectomes do really well in general, better than basically all the types of models, except for these using a receptor similarity connectome. So what is this? So this is sort of the distribution of receptors in a given region. And so when we look at receptor covariance, we're basically saying that brain regions that have a very similar neuroreceptor profile to our epicenter, the entorhinal cortex, those show tau sooner. And when we have regions that have a very, very different sort of receptor profile than entorhinal cortex, it shows tau later. That's the sort of idea behind that. And we thought that was very compelling and interesting because it's not something that tends to be studied very carefully in Alzheimer's disease compared to, I think, other diseases. And so we wanted to know, what is that receptor profile that sort of, tau seems to follow. So we have all of these different PET measures of different neuroreceptors from the serotonin system, the dopaminergic system, and so forth. And we just said, we just correlated the overall kind of tau sequence and tau load with these different receptor maps. And so again, this is a very busy slide. I'm going to actually skip through a bunch of this because I want to get to a certain point that I think it might be interesting to people here. We noticed that the sort of GABA receptors are sort of anti-correlated with the overall distribution of tau. And that's interesting because there's a lot of interesting work going on about the excitatory inhibitory ratio of different parts of the brain and Alzheimer's disease. And so we got together with some colleagues at Jülich who are kind of experts in this, and we sort of got together these ratios, excitatory inhibitory receptor ratios, and looked if they were correlated with the overall progression of tau. And we saw that they really were, and particularly we found this sort of ionotropic glutamate to GABA ratio was pretty positively correlated with tau pathology. And it's very interesting to us because it really means that as tau progresses through the brain, it sort of progresses along this it starts in areas that have a really high excitatory tone and basically spreads across regions with a decreasing excitatory tone over time. And so I think this is very interesting to us. And there's a number of reasons that's interesting. Basically, there's been some really nice work in the animal literature that has shown that depending on who you talk to, both amyloid and tau actually depolarize neurons when they're nearby them. And that's really problematic because when neurons depolarize, apparently they also secrete both amyloid and tau. And so there's a lot of people that think that there's this kind of vicious cycle that goes on where basically, pathology is being secreted. That pathology then leads to hyperactivity of neurons, and this creates this kind of hyperactivity of a circuit that can then spread along to other circuits. And so that's something that we're trying to basically pursue this a little bit further and build this excitatory and inhibitory information into our models. Okay, so that's kind of it for that kind of first half of the talk. So basically, we're using these connectome-based models to try to learn something about the disease that we didn't already know, and we can use that to basically go to our colleagues who are, more experimentalists and who are working with, animals and so forth, and try to test these hypotheses as much as we can. But we like that what we're doing is really based in human data, which is important because it's humans who are actually getting these diseases. And that paper is just dropped on bioRxiv if people are interested in that, like last week. So hopefully that will move forward. But we don't want to stop with just biological, insights, right? We also want to. A lot of the people that we work with are clinicians, and they're of course interested in the how the neuropathology works, but ultimately they see a lot of patients and they want to help their patients, not just by finding cures to the disease, but also by building tools that help in terms of biomarkers and in terms of clinical trials and so forth, right? And so the question is, can we use these types of models to actually, learn something about an individual patient to predict their future or to do something that would be helpful in a clinical trial? So this is one of the other things that we want to do with connectome-based models. We want to be able to make individual level predictions. So I'm going to talk to you about a sort of different connectome-based model that we've been working on with this really awesome, he's now a postdoc at Lund University named Pavan Shagar, but he did his PhD at Oxford. And we sort of started this project when he was in Oxford, and we've been continuing it now. But it's another connectome-based model, a similar idea, right? You're diffusing, your pathology through the brain's network measured with structural connectivity. But we're just adding some very simple sort of parameters into this model, where first we're allowing sort of brain regions to increase in amyloid beta, and that increase is really dependent on the sort of carrying capacity of a region, which is just how much pathology eventually it's able to support, that brain region's able to support. And then we have these terms related to tau. So there's this sort of age-related accumulation that happens in everyone, and then there's this axonal spreading of tau that happens, and there's this sort of interaction between A beta and tau, which causes increased production of tau. And of course, we're interested in brain atrophy because that's what sort of leads to cognitive decline, right? Just having pathology doesn't do anything. You actually have death of neurons, and it's the death of neurons that leads to, decrements in cognition. And so this is actually a very, very simple model if you look at it, but it captures kind of the main elements, mechanistic elements of macro scale interactions between these brain regions, between these different neuropathologies. And so we can, this is a sort of simulation of this model, and in all of the models that I've shown you, you can basically display them like this. But I think this one's nice because you're getting all three elements of Alzheimer's disease: the amyloid, the tau, and the neurodegeneration. So I'm going to play this simulation, and what you're going to see is first, early on, this sort of saturation of amyloid beta that leads slowly to the accumulation of tau, specifically in Alzheimer's-like regions, and eventually that also leads to neurodegeneration in the same regions. And so it's pretty interesting. It sort of plays out exactly like we would want it to. But what's interesting is that we don't actually tell the model anything about where, we don't really give it much information about where tau is going to build up. This is really, or neurodegeneration for that matter. It's really based on two things: the amount of amyloid in a brain region and spread through neuronal networks. And the sort of, very, very Alzheimer's-like kind of load of tau that we see in the brain here is really an emergent property of this model. And that's great because it means we're with just a few parameters, we're doing a pretty good job simulating the spread of all three tenets of the Alzheimer's disease spectrum. And so the question is, can you fit this model to real data? And if you do, how does it look? It looks pretty good. So this is actually change in amyloid, tau, and atrophy predicted on the y-axis, observed on the x-axis, and again, each dot is a brain region, and the red regions are the regions that are the primary Alzheimer's regions that we're really most interested in. And so when you fit this model to real data, you do actually do a really good job basically at an individual level and also at a regional level predicting how fast pathology is going to build up. So this is cool, and so we're kind of trying to push this into a sphere where it can be useful for clinicians. But really, what everyone's interested in now is clinical trials for tau pathology, and people want to be able to know what's going to happen in a patient. And so we've been collaborating with some people at Roche that have a sort of drug that they're giving people, and this drug lowers amyloid beta. And in theory, if you lower amyloid beta, this will sort of reduce the sort of mechanistic interaction between amyloid and tau, and will lower tau as well, and degeneration won't happen. And so this from mouse models, we know that this drug sort of enters the brain through the CSF. So what we can do is simulate this treatment coming into the CSF and then diffusing through the brain, and we can and basically see what happens to amyloid and tau, whether you have a placebo or the drug. So I'll play these two simulations. You can see both. You can see here there's amyloid building up, but here no amyloid. It starts to go down. And here you see tau and neurodegeneration start to happen. And with the drug, because you have the drug, we see less buildup of amyloid and tau. Okay, not that surprising, right? That's sort of how we drew it up. We have these interactions built into the model. So if you remove the amyloid, you lower the interactions, and you're going to see less of the pathology in the brain. But we can still use this to be useful, right? So one of the things that we did is we have this time period in months where we have someone who's a random person who is on the Alzheimer's disease spectrum. Maybe they're very early, maybe they're very late. That's kind of represented by this kind of time course. And the 0 is basically when they have positive amyloid in the brain. And so if we kind of can do these simulations at different time points and just see, if we start at different times, how much of an effect does it have on the downstream tau or atrophy in a given patient? And when we do that, we see that if you start sort of earlier, at the endpoint you have a lot less pathology. But if you start too late, at the endpoint you still end up having quite a bit of pathology. And so this is actually kind of useful information to us because it really suggests that one wants to start these treatments early on in the disease and not later. Again, seems self-evident, but it's nice to have some actually information you can point to. And so we actually ended up doing this across many, many time points, not just these 4. And what we noticed is that if you look at sort of the difference in the efficacy in lowering brain atrophy and lowering brain tau over time, what you see is there actually ends up being a time point where you are maximizing the efficacy of giving this drug. And really, if you go past that time point, I don't want to say it's too late, but you're really, really getting reduced efficacy of giving this drug. And so we can sort of, take a given person who's coming into a clinic and say, are you before this time point? If so, we should give you this drug. If not, we should probably be giving you a different type of drug that targets, something downstream. All right, so that's kind of that's a sort of conclusion of these connectome-based models. So really the idea is that we're using brain networks as a tool to support simulations of neurodegenerative diseases, and we're trying to use these to learn things about the disease and also to get some sort of clinical benefit. So it's still early days in a lot of this, but we're trying to push it, and we think it's pretty interesting and useful. And let me just check the time. I have how much time do I have?
5 minutes.
Okay, then I'll just end it there, and we can skip the last step. Maybe I'll go to my ending slide, my thank you slide, sorry. And yeah, thank you.